Hi, welcome once again to our YouTube channel. Thanks for subscribing to our channel, really appreciate you. In this particular tutorial video, we want to talk about the biology practical specimen for the Waec 2025 examination. The specimens that Waec has chosen to use or to represent for this year's 2025 Waec biology practical. Those are the things I want to look at today. So we're talking about the characteristics, we talk about so many properties of this, we talk about possible uses, possible questions that you might be coming across in the course of the examination. So without wasting too much of our time, we'll go straight into listing all of these specimens one after the other as they have been itemized by Waec. So starting from our specimen A, specimen A, what we have there is bean weevil. Bean weevil. And our specimen B, we have bean seed. Bean seed with holes, bean seeds that have been perforated by pests. And then the third one there, our specimen C, is water leaf. Water leaf is our specimen C, the water leaf plant. The whole water leaf plant is our specimen C. And our specimen D, we have it to be quill feather. Quill feather of a domestic fowl. Quill feather of a domestic fowl. And our specimen E, we have it to be the pineapple fruit. The pineapple fruit. That is what we have our specimen. That is what we have our specimen E to be. Now for the pineapple fruit, we'll be looking at the longitudinal section. Longitudinal section. And then for our specimen F, what we have there is going to be orange fruits. Orange fruits. And for this one, we'll be having the transverse cut or the transverse section. The transverse section. And for our specimen G now, what we have for our specimen G is raw egg of a domestic fowl. Raw egg of a domestic fowl. Raw egg of a domestic fowl that has been freshly procured. Freshly procured with shell. With shell intact. With shell intact. That is our specimen G. And our specimen H, what we have our specimen H is the test tube containing 3 ml of water. Test tube containing 3 ml of water, which has been placed on a test tube rack. That's our specimen H. And our specimen J now, which are longitudinal section of boiled fish with shell. So these are the list of specimens that Waec has given to us this year to use as biology practical experiment, biology practical examination. Specimen A, we have bean weevil. Specimen B, we have bean seed with holes. Specimen C, we have water leaf plant, the whole water leaf plant. And for our specimen D, we have the quill feather of a domestic fowl. Specimen E is pineapple fruit, the longitudinal section of a pineapple fruit, while specimen F is the orange fruit, the transverse section of the orange fruit. Specimen G is raw egg of a domestic fowl that has been freshly procured with shell intact. And a specimen H is test tube containing 3 mils of water that has been placed on a test tube rack, while specimen J is the longitudinal section of boiled fish with its shell intact. So without wasting too much of our time, we'll go straight into talking about each of these specimens as we see them one after the other. So for our specimen A, specimen A, we have the bean weevil. So what do you want to say about the bean weevil? First and foremost, we will have to talk about its classification. So the classification for the bean weevil now, the kingdom, we have it to be Animalia. The phylum, we have it to be Arthropoda. We have the class to be Insecta. We have the order to be Coleoptera. We have the family to be the Chrysomelidae. Chrysomelidae. We have the genus to be the Acanthocelides. Acanthocelides. And then we have the species to be the Optectus. Optectus. So with this now, we can actually write out the scientific name of this organism to be, we have it to be the Acanthocelides Optectus. Acanthocelides Optectus. That is the scientific name of this organism. And when we write the scientific name, we don't join the underline. You can see how we have done that. Let me use, when we write the scientific name, we don't join the underline. You can see how I've written this. So that is how it is written. You put them apart, you don't join them, and you make sure that the first letter is written in capital letters, while every other letters come in lowercase or in small letters. So that is the scientific name of this particular organism, and it happens to be Acanthocelides Optectus. Now, what do you want to say about this organism? This organism, they primarily feed on common beans. Primarily feed on the common beans. That is what they feed on. And now it is not only the beans, they also feed on peas and other legumes and other leguminous plants and other leguminous plants. Those are the things that they feed on. They are pests of legume seeds, but they do not only feed on the seeds, they also feed on the plants while the plants are on the field. They are pests of legumes that feed also on the plants while on the field, as well as on the seeds, as well as on the seeds while in storage, as well as feeding on the seeds while the seeds are in storage. But it should be noted that it's only the larva that actually feeds on the seeds. Only the larva feeds on seeds. It is the adults now, adults that feed on the plants. They feed on the pollens. Adults feed on pollens. Adults, they feed on the pollens of the plants on the field. They feed on the pollens of the plant on the field. Now, a single mature female bean weevil can actually lay up to 300 eggs per time. Single mature female bean weevil can lay up to 300 eggs. It can lay up to 300 eggs. Now, for this particular crop, they have their own metamorphosis to be complete. Their metamorphosis is complete. They have complete metamorphosis. So if they have complete metamorphosis, it means it looks this way: from egg to larva and then to pupa before it goes to the adult stage. So that is how this particular organism is. Now, when we talk about economic importance of this organism, economic importance of this organism, we should be ready to make it clear that they have their own economic importance, but the economic importance is destructive. So one, they feed on and destroy seeds. Yes, they feed on and they destroy seeds. That is their major economic importance. So that is what we have to say about the bean weevil. Let me show us a picture of what the bean weevil looks like. You can see this is an example of a bean weevil. You can see how the picture looks. That is what the bean weevil is. So it has a pair of antenna. It has its legs. It has 3 pairs of legs. You can see it has its head here. This is where the head is, and then it has a prothorax as well as what the abdominal part of the body. So that is what that particular organism is looking like. This is another picture of the Acanthocelides obtectors. You can see this as well. You can see that, as well as this. We have this here as a labeled diagram of that particular substance, of that particular organism. You can see, so I said earlier on that as its head, you can see this is the head carrying a pair of antenna. You can see the pair of antenna. It has its mouth parts, the mandible and the maxillae, and then on the prothorax here we have it having the first limbs, but it has a total of 3 pairs of limbs. So that is what that particular organism is looking like. So moving to this next specimen now, our specimen B. Our specimen B is the bean seed with holes. bean seed with holes. Bean seed with holes. That is our specimen B. So what do we have to say about our specimen B, the bean seed that has holes? First and foremost, we talk about their scientific classification. So we start from this. The kingdom, it is Plantae, because it is a plant, and then the phylum, it is Tracheophyta. Tracheophyta. We have the class to be Angiosperma. Angiosperma. The order for this particular crop is Fabales, and then the family to which it belongs is the Fabaceae. Fabaceae. The genus is the Phaseolus. Phaseolus, and the species to it is the Vulgaris. The Vulgaris. That is the species to this particular plant. So that is how it is being classified. So let's move further. There are different types of bean, but we just base our case study here on bean with holes. That is just what we are talking about. So we just generalize now. Beans are creepers or climbers. Beans are legumes. Beans are legumes. We also call them pulses, that are creepers or climbers. Yes, we call them creepers or climbers because they have the ability to actually grow, and they do not, because they are vines, they do not have the ability to grow a strong stem. Since they do not have the ability to grow strong stem, they cannot stand upright. So they creep on the ground, they creep on the earth surface. That is why we call them creepers. And if they are opportune to grow along or grow in an environment where there is possibility of having a pole or a stick, something like a stalk, they can actually creep on and wind themselves around that pole to climb up on that pole. So that is also the reason why we call them climbers. So we have different varieties of this particular crop. Varieties. Example, we have the kidney bean. We have the kidney bean, we have the black bean, we have the pinto bean, we have the navy bean, etc. These are so many examples of the varieties of the bean. Now, beans are summer crops that need worms to grow. They are summer crops that needs worms to grow. Yes, they need worms to grow, and it is capable of nitrogen fixation. They are capable of nitrogen fixation, and hence they are usually not requiring any form of fertilizer. Hence they need less fertilizer. Hence they need less fertilizer because they actually have the ability to provide the common material. They are basically called vines. Yes, I said that earlier on. Basically called vines, and so they need external supports. So they need external supports. They need external supports like a cane or a pole. Like cane or pole. Yes, they need a cane or a pole to wind about. It is an important source of protein. is an important source of protein. For this particular crop now, we can talk about its placentation. The placentation for it is marginal. Placentation is marginal. It has all its seeds arranged, or the seeds arranged in the pod serially. So it is marginal, and the propagation for it is propagated vegetatively by seed. Vegetatively by seeds. That is how it is being propagated. While the maturity for this particular crop, or maturity period, it takes about 2 months. 2 months from planting. That is how it takes this crop for it to mature into a full-grown crop that is ready to be harvested. So that is about the bean seed. But particularly what we are talking about now for this specimen is bean seed with holes. So there are crops that have been eaten up by bugs, by the weevil that we talked about in our specimen A. So these are examples of this bean. You can see this bean now, the bean seed here. You can see the weevil on it, and it has eaten up the bean. It has bored holes on the seed. That is what we are talking about. Let me show us another example. You can see here now, you can see some of these bean seeds with holes. They have been eaten up by bugs. You can see them, that they have been eaten up by bugs. You can see the weevil on it, and so they have created holes on the seed. So that's why we said that the economic importance of this bean weevil is that they destroy seeds. So this is another pictorial representation of what we are talking about. You can see all of these seeds, all of these bean seeds here, they have been perforated, and these are done on them by the bean weevil that you are seeing on them. So that is that about bean seed with holes. So we should not forget that the scientific name for this particular bean, for the bean that we have talked about, the scientific name is the Phaseolus vulgaris. That is the scientific name of this particular crop, Phaseolus vulgaris. So don't forget, you don't join the underline under any circumstance. So our next specimen is Specimen C, which is water leaf. Water leaf, the whole, okay, water leaf plants. Water leaf plants. The complete water leaf plant, that is what our next specimen is. So let's quickly see what we have to say about the specimen. First and foremost, we'll talk about its classification. So it belongs to the kingdom Plantae. The phylum there, it belongs to the Trachyphyta. The class, it is a Magnoliopsida. Magnoliopsida. And then we have its order to be the Caryophyllalis. The Caryophyllalis, we have the family to be the Thalinaceae. Thalinaceae, the genus is Stalinum, and the species is the Triangulare. So with this now, we can actually say that its scientific name, its botanical name, is going to be the Talinum triangulare. Talinum triangulare, that is the scientific name or the botanical name for this particular crop. So what do you need to know about this crop? It is called water leaf because of its high water retention. It is called water leaf because of its high water retention. Yes, it has very high amount of water in its stem. That is why they adopted that name, water leaf, and it is commonly found in West and Central Africa. Commonly found in West and Central Africa. That is where you find this particular crop. They are known for their medicinal purposes and are used traditionally for medicine, for treatment of various ailments. Known for its medicinal properties and used in the treatment of many ailments. Yes, it has very important properties like it has antioxidant properties, it has antioxidant properties, it has anti-inflammatory properties, as well as anti-diabetic properties. So these properties are very essential, and those are some of the reasons why they have been very cultivated, or why they are very important. It is a good source of vitamin, like we said. It is a good source of vitamin and minerals. Consuming this particular crop enriches you with high amount of vitamins and minerals. It can be cultivated or propagated using tiny seeds that it produces. It is usually cultivated or propagated. Usually cultivated or propagated using tiny seeds it produces. So these are some of the things that we need to say about the Talinum triangulare. So let me show us a picture of what we are talking about. This is the Talinum triangulare. You can see it here. You can see the picture here. That is what it looks like. That is the water leaf. So it is here in this flowery part that we have the seeds being born, and so when the seeds fall on a suitable substrate, it actually produces or develops into a new plant that is that gets matured. So this is another example. You can see a potted or a whole plant of that Talinum triangulare. You can see this is a labeled diagram of that particular crop that we're talking about. We can see here now that it has the shoot everything that is above the soil level here like this fall to the shoot system, while everything that is underneath the soil or in the soil is in the root system. So we have the primary and the secondary root. And then on the stalk here now, we now have the flowers, we have the fruits, the stem, we have the leaves, we have the node and the antinode, the bud, etc. So this is the label diagram of the Talinum triangulare. So our next specimen now is our specimen D. Specimen D, which is the quill feather of a domestic fowl. Quill feather of a domestic fowl. Since it is the quill feather of a domestic fowl, we can quickly talk about the classification of that particular organism. Though we can get quill feather from other types of poultry beds, but we are specifically talking about that of the domestic fowl now. So when we look at the domestic fowl, this is how the classification goes. It belongs to the kingdom Animalia. The phylum there is the Chordata, because it is a chordate. It has backbone. We have a class to be aves. And then the order there, we have it to be the Galliforms. We have it to become the Galliformes. And then the family, it is the Phasinidae. The genus, we call it Gallus. And then the species is Domestica. That is why scientifically we refer to it as the Gallus domestica. Gallus domestica. Some textbooks you see it written as Gallus species. So remember, this is how the scientific name is being written. Alright, so what are we saying about the quill feather? This is the larger or stiff feather from the birds. A large stiff feather from the birds wings or tail. So these are the regions that we actually find these particular types of feather called the quill feather. You find them in the wings and in the tail. So they are usually referred to as flight feathers. They are hollow. And they are tube-like. They are hollow and tube-like. Because they have holes, they are very, very light, and that is why they are referred to as flight feathers. Hence, called or referred to as flight feathers. They are referred to as flight feathers. The quill or the calamus is a part of the feather. The quill, or we also call it the calamus, I will show that to us shortly, is a part of the feather that attaches to the bed. Is the part of the feather that attaches to the bird's skin. It attaches to the bird's skin, and the rachis which is the shaft of the feather, which is the shaft of the feather, extends from the quill. extends from the quill. Now historically, this particular specimen that we're talking about have been said to be used as a writing instrument. They used it as writing material in time past. So historically, it was used as a writing tool before the intervention of modern pens. Historically, it was used as a writing tool depth in ink. They usually put it into ink before the emergence of what we know today as pen. Before the emergence of pen. So that is about the quill feather. This is what the quill feather looks like. You can see it. That's a picture of the quill feather. So this is what we refer to as the calamus. This tip here is what we refer to as the calamus. So that is the part that attaches itself to the animal's skin. That part is attached to the animal skin, and then we have that rachis. Now all of this place here called the rachis, and that is what carries the barbs of the feather. You can see it carries the barb of the feather. All of these tiny feathery materials here, they are what we refer to as the barb. So the calamus extends into the rachis that eventually carries the barbs. So that is what the quill feather looks like. This is another picture, a labeled diagram of the quill. You can see. So this part is what we refer to as the calamus that is attached into the skin of the bird. So this particular species is usually found either on the wings or on the tails of the birds. Now, in case you are told to talk about quill feather and then relate it with palm front, for example, you are said to talk about similarities between those two. Few of the similarities that you can make mention of, let me just put that here. Similarities between palm front and quill feather. One is that they both have rachis. They both have rachis, and then they both have barbs. They both have rachis and barbs. This is what I'm saying here. They have rachis and barbs. If you look at the palm front, it has something similar to this here, and then it has these ones here that are somewhat like the leaves. So they both have the veins or the rachis, and then they have the barbs. So that's the major similarity that you can have between the quill feather and palm front. Peradventure, you are told to talk about some similarities between them. So the next specimen we have there is our specimen E. Specimen E, which is the pineapple fruits. Pineapple fruits. And this time around we're talking about the longitudinal section. The longitudinal section of the pineapple fruits. That is what we are looking at. So, like we have done in the past, we quickly look at the classification of this particular crop. So, classifying this crop now, it belongs to the kingdom Plantae. Kingdom Plantae, and then the phylum there is the Trachyphyta. Trachyphyta. The class to which it belongs is the Angiosperma. Angiosperma. Then we have the order to be Poalis. For this, we have the phylum to be the Bromeliaceae. And then we have the genus to be Ananas. And the species is called Comosus. The species is called Comosus. And with this now, we can deduce the scientific name of this species to be the Ananas Comosus. Ananas Comosus. That is the scientific name. So that is that about classifying the organism. So let's look at the crop. It is a large tropical fruit with spiky tough skin. Pineapple is a large tropical fruit with spiky tough skin. With spiky tough skin. That is what this particular organism is. It has tough skin with sweet internal parts. It is highly rich in vitamin C. Highly rich in vitamin C. And that is why it has antioxidant properties. It aids digestion. And enhances weight loss. Yes, those are some of the properties or some of the effects of this particular crop. It is a sexual stimulant as well. It is a sexual stimulant. It is rich in antioxidant properties. Yes, that's why I said it is. Okay, let me just put it here. It has antioxidant properties. Also, it produces hormones responsible for skin and tissue healing. Also produces hormones responsible for skin and tissue healing. So it can help to put the skin in a very, very good condition. It is first a fruit, and then we call it first fruit, after which it is seen as a compound fruit. That is how it is being classified. This particular crop has a placentation which is axial. Placentation is axial. That means it has its oval centrally placed. The oval is centrally attached, and then we say that this particular crop is propagated by sucker. It's propagated vegetatively. Propagation vegetatively. vegetatively using its suckers. That is how it is propagated. So this is what we are talking about. This is an example of a pineapple fruit. You can see it. That is what the pineapple fruit looks like. So these are other pictures of the pineapple fruit. This is the pineapple fruit while it is still on its stalk. You can see. So we are looking at the longitudinal cut of the pineapple fruit. So this is what the cut will look like when it is cut from top to bottom. Midway, that is what the cut will look like. So that's the longitudinal cut of a pineapple fruit. So this is the labeled diagram of that crop. When you cut it longitudinally, we find out that in the middle here we will have what is called the core, and close to the core we have a fleshy part that is eaten. You can see that, and on the top here we have some of the leaves of that particular plant, so to say. So that is what this particular crop is about. So we move to the next specimen now, which is our specimen F. Specimen F, which happens to be orange fruit. Orange fruit. And what are we discussing about the orange fruit? We are looking at the transverse section. Transverse section of the orange fruit. So as we have done in the past, we will quickly make mention of the classification of this particular crop. So classifying it now, we have it belonging to the class, the kingdom Pantae, and then the phylum. The phylum is, we have it to be the Magnoliophyta. Magnoliophyta. We have a class to be Dicotyledon. Dicotyledon. And then we have the order to be Sapindalis. We have the order to be Sapindalis. The family there, we have it to be the Rutaceae. Rutaceae. The genus is Citrus. And then the species is Sinensis. The species is Sinensis. So with this now, we can actually write out its scientific name to be the Citrus Sinensis. Citrus Sinensis, that is the scientific name of this particular specimen. So you don't forget how it is written. And then we talk about the organism now. Looking at it now, we refer to it as a spherical. Okay, let me just describe it first. It has a waxy skin. Orange has a waxy skin. It is spherical with outer peel. It is a spherical fruit. It is a spherical fruit. With outer peel. The outer peel is what we refer to as the epicap. With underlining fibrous layer. With underlining fibrous layer. This underlining fibrous layer is the part that we refer to as the mesocap. I will show these things to us shortly. I will show us all what we are talking about now. And then it has the juicy part called the endocap. And the juicy part that we actually munch on or take out to suck out, the juice that we suck out, the juicy part called the endocap. So those are the classification, the parts of this particular fruit. It undergoes asexual reproduction. Undergoes asexual reproduction. So when we talk about how it is being propagated, we know we propagate this particular organism, though by grafting. Sometimes we actually do grafting. Okay, let me put that here. Grafting, or sometimes by budding. Now, it is not that it doesn't produce seeds that can be used, but oftentimes the seeds do not actually grow as expected. So it is usually grafted from an existing plant, or grafted or budded. That is how this particular crop is being cultivated. The placentation of this crop, is axile. Placentation is axile. So that means the oval is centrally attached. Now, looking at it, what kind of fruit it is it? Let me put that here. It is a fruit, and then we refer to it as true fruit. True fruit, and then because it is found from ovary. Because it is found from ovary, that's why we refer to it as the true fruit. Then it is also known as a simple fruit. A simple fruit. Let me put that here. Monocarpous. After which we see it as a succulent. It is seen as a succulent. And lastly, it falls under the Hesperidium. Hesperidium. So this is how the classification of this particular organism is. It is a true fruit. It's a true fruit, because it is from the ovary, and it belongs to the simple monocarpous fruit, a succulent fruit, as well as an Hesperidium. So that is the classification of this organism. So the transverse cut of the organism is what we want to look at now. This is what orange looks like. You can see that is what orange looks like. You can see them in the background. So when it is cut in a transverse section, that is cutting it from left to right. If you cut it from left to right, this is what the cut will be looking like, so that we'll be able to, as we said earlier on, that it is called the epicap. This part here is called the epicap, and then we have this part here that is white in color. It is called the mesocap. That is the mesocap. This part that is orange is called the epicap, and then we have the white part, which is called the mesocap, and the inner part, the juicy part here, which is called the endocap. So that is about the oranges. So we move to the next specimen now, which is Specimen G. Specimen G, and our Specimen G is the raw egg of a domestic fowl. Raw egg of a domestic fowl. That is our Specimen G. So we have done the classification previously when we talked about it in Specimen D. So from that now we can write out the scientific name of this particular organism. So we said it is called the Gallus domestica. You shouldn't forget that, but you can actually refer back to our Specimen D to be able to understand or to identify how the classification is. So that is just the scientific name there. So what do you have to say about this particular thing? The eggs are gotten from domestic fowls. eggs are gotten from domestic fowls. They are gotten from domestic fowls or birds. They are gotten from domestic fowls or birds, which could be layers or broilers. Which could be layers or broilers. It could be layers, it could also be broilers. The birds are oviparous, that is why they have the ability to lay eggs. Birds are oviparous, so they lay their eggs. They do not give birth to their young ones alive, so they usually lay eggs. Now the egg contains albumin, which is a clear liquid. It contains albumin, a clear liquid that turns white after heating. That turns white after heating. Yes, when you apply heat, when you apply heat to the egg, you notice that the liquid part of the egg, which is called the albumin, starts to solidify, and as it solidifies, it starts to take a white coloration form. Then it also contains water and protein, serving as nutrients. It also contains water and protein serving as nutrients. Yes, that is very, very essential. It also it is very, very rich in vitamin, so many vitamins like vitamin A, rich in vitamins A, vitamin D, vitamin E, vitamin K. We also have all these vitamins here, they are referred to as fat soluble vitamin. And then we also have it containing the water soluble vitamins like vitamin B1, it has vitamin B2, vitamin B5, vitamin B6, vitamin B9, vitamin B12, which is cyanocobalamin. All of these are very essential vitamins that are found in the egg. Now it also has a yolk. The yolk contains carbohydrates. The yolk contains carbohydrates. The yolk contains carbohydrates and the genetic energy. It generates energy for the embryo and generates energy for embryo. So the energy that is used by the embryo is generated by the yolk. It deepens to yellow. The yolk deepens to yellow color at the center after heating. Yolk deepens to yellow coloration, to yellow in the center after heat has been applied to it. So the color changes to yellow. Looking at it now, the eggshell itself has cancerous nature, which gives it or provides mechanical protection for the contents of the egg. The eggshell has cancerous nature, which gives or provides mechanical protection to content of the cell, which gives mechanical support or protection to the contents in the shell or to the contents of the egg. The shell is porous for gaseous exchange. The shell is porous for gaseous exchange, and with the environment, the shell permits gases into the egg for gaseous exchange, and it houses and contains the embryo. There are different colors of these eggs. Okay, let me just put that here. The shell thereby permitting gaseous exchange, thereby permitting gaseous exchange. Yes, it permits gaseous exchange. Incubation is about 21 days. Incubation is 21 days. Let me put that here. Incubation is 21 days. So if the egg is to be hatched, it will take 21 days. We have different colors of the egg. The colors, we could have it to be white. Also, we could have it to be brown. We have white, we have brown. Now, the egg candler, if you have to check the viability of the egg, we make use of a particular instrument called the egg candler. Egg candler can be used to check the viability of the egg. We can use egg candler to check the viability of the egg. So these are some of the things that we need to know about the egg as it is. So this is what the egg looks like. You can see this is an egg, the brown egg. We have this one as well. You can see that is what the egg looks like. You can see a mixture of brown and white eggs here. Now this one here has brown shells, and then we have this one here having white shells. So these are the things that we refer to as the eggs. So we move to the next specimen now, which is specimen H. Specimen H, and our specimen H is test tube containing 3 ml of water. Specimen H is test tube containing 3 ml of water, and the water is placed on a test tube rack. So what do we have to say about the test tube that contains water? Specifically, what we want to look at now, we'll be looking at the content of the test tube. That is what we want to look at now, the content of the test tube. So water, what is water? We have it's chemically, the formula is chemically H2O. Water is composed of hydrogen and oxygen. That is why chemically it is written as H2O. That is what we refer to water as. Now, water has its own uses. Uses: 1. It is used domestically for cooking, washing, drinking, etc. It is used in agriculture to help water the plants, Three, we can say that water, when it falls as rain, helps to increase moisture, to increase humidity. It helps to increase humidity. If you look at the properties of water, we say that water is colorless, odorless, and tasteless. These are some of the properties of the constituent elements that make up water as it is. Hydrogen and oxygen, they are colorless, odorless, and tasteless, and that is why water has adopted that particular property of theirs. So these are some of the things that we can use water as. Now, water is seen water is a polar solvent. It is also a neutral substance. Because it has a pH of exactly 7.0. A pH of 7.0. So these are some of the things that we need to know about water in this experiment. So this is water in a test tube. You can see the test tube having water in it, and then on a test tube rack. You can see it here, water in the test tube rack. That is what this particular experiment is talking about. Now we're going to talk about our last specimen, which is Specimen J. Specimen J, and that is the longitudinal section of boiled egg. Longitudinal section of boiled egg. With shell. Longitudinal section of boiled egg with shell. That is our last specimen there. So we will not be saying much about this particular setup, because everything we have discussed while we discussed Specimen G here, everything we talked about Specimen G here is what we will still need to be talking about the longitudinal section of boiled egg. So I'll just show us what the diagram of that particular experiment will be, the specimen. So this is what the diagram will look like. You can see the diagram now. First and foremost, let me start with this. This is how the longitudinal section will look like of the egg. So it is cut from top to bottom. So we'll be having it showing the shell. You can see the shell which is intact. So when it is cut longitudinally, we can see that from the top of the shell now we have something that looks like a rope that spirals down towards the middle where we have the yolk. That thing is called the chalazae. The chalazae, it is found above and beneath the yolk. It holds the yolk centrally in place in the egg. And then we have the yolk here, which is we have this white part here. This white part here is what we refer to as the albumin. The white part is what we refer to as the albumin. And then we have this place here to be the yolk. We have this place here to be the yolk, and towards the middle here of the yolk we have what is called the germinal disc. That's the blastoderm. It's called the germinal disc. And then underneath the yolk again, like I said, we have the chalazae up and beneath, and then under that we have the air space. You can see that. So this is what that diagram that we have just talked about is. So this part here is where we have the chalazae, and then we have the yolk, we have the blastoderm, we have the chalazae underneath here, and then the air space here. The outermost covering is what we refer to as the shell. So we can see that here. We can also see another diagram here, a labeled diagram of what we have talked about. So that is about specimen J. So that is as far as we can come for the biology practical experiments or biology practical session, talking about all of the specimens that Waec has chosen to use for this year's Waec 2025 biology practical examination. I will encourage us to please try as much as possible to go through all of this that has been discussed in this tutorial video again, to familiarize yourself with all of the specimens that you have talked about. Know each of the specimens, their characteristic properties, their uses, how they are formed, their classification, and the rest of those. I wish you the best of luck in your examination. For those of us that are yet to subscribe to our channel, please subscribe to our channel. Don't fail to subscribe. Subscribing to our channel has a whole lot of benefits to offer you. When you subscribe, don't forget to put on the notification button so as to be notified of subsequent uploads when we put them up on the channel. We will still be having some videos to discuss about some specimens in the examination that is going on, talking about agric. We have done that of animal husbandry. If you are still looking for that, you can check our channel, and that of chemistry will still be coming up. Please like our content, share them with your friends, let them also benefit from what you are getting, and we want to hear from you. So please do not forget, do not hesitate to put your questions down in our comment section, and we will try as much as possible to answer you promptly on your burning questions. Thank you very much, and bye for now.